All right. Well, I think since Jack, you've presented many times before, I won't really do an introduction or anything, but so I'll let you just get started.
That's good. Thanks, Sergey. All right, I think I'm sharing my screen, and then somebody can tell me if I'm not. Sorry. Okay, that was a testing 1, 2, 3. I've been working lately on, well, I've been working for years on graph neural networks, but lately I've been working on using Rosetta as a companion as much as possible. Just, I think all of us over the past few years have really been trying to see what is Rosetta's role. To what extent is Rosetta helpful in the machine learning wave? And every few months, there always seems to be new ways that there's like compatibility between fields. And so this, the title is kind of broad and it's not really unique. Like Chengpeng already shared months or maybe even years ago now that, sorry, this is Chengpeng from the Car Lab, where they used Rosetta terms as features for graph neural networks, nodes and edges, and showed that it performed really well. And while she was doing that, we were doing the same thing in Menton and really came to similar conclusions. So in these slides today, I'm going to share how it works for us and why we think this is so successful. And then there's a little bit of a bit at the end, like how can we improve Rosetta to make quality of life as a companion tool better. So the first 2 slides are going to be setting up the use case. And this is for the sake of having a narrative and like sharing our data and results. If it doesn't grab you as interesting, it won't take up the whole meeting. But I'll just dive in for a bit at least. And mention we work a lot with cyclic peptides. And as like we shared in a lot of like Vikram's conference talks and from other labs as well in Rosetta, a big part of designing cyclic peptides, especially as binders, is not necessarily sequence optimization, but validation of a design. You really want a high fold propensity. And so this is cyclic peptide on the left. What you'd really like is, given a bound state of your cyclic peptide to a bound target, you'd really like that cyclic peptide to adopt a similar conformation in the unbound state so that it doesn't have to change conformation a lot when, in order to bind, so the energy barrier of binding is lower. From a data perspective, we like to view it in these folding funnels, which I think a lot of us are familiar with, but for those who aren't, these show a lot of different conformations of the same sequence. The x-axis is RMSD from your cyclic peptide binding, your conformation in the bound state, so your desired state that you're measuring the fold propensity for. And then the y-axis is the energy. Ideally, when the RMSD is very low, the energy is also low. And then as you deviate more and more from your designed conformation, energy increases. So this funnel on the left is exactly what we'd like to see. I stole this from one of Shannon Smith's papers, but we see this pattern a lot in our community's literature. And on the right, this is a less ideal. Some might say bad, though it's largely a spectrum. Because here, as you deviate from your 0 RMSD state, there's actually a more stable conformation elsewhere. So in broad strokes, the question is, given this conformation, is there more likely to be a more stable conformation for the same sequence? And we'd really like that answer to be no, but it's also very important to be able to predict that. I really like this figure from Chris Norton's paper last year. So I did try and include it whenever I can. Especially when I was trained in Rosetta, our goal when designing sequence was minimize the energy for your conformation. But this is really more of it's a more complicated problem than that. So I'm not on this paper, but I really love this figure. And what they've shown is that, if you have, one-dimensional conformation landscape and two possible sequences, maybe this point on the conformation landscape is your desired conformation. Sequence A is always a worse like Rosetta energy than sequence B. However, by far its global minimum is at the desired conformation. Where sequence B, it actually has a lower energy at your desired conformation. However, it's not the global minimum, and there is a competing state. So even though the conventional, minimize my energy at my conformation technique would select sequence B if you're designing sequence optimization at for this conformation. In a lot of cases, sequence A is actually the preferable sequence. This is really just the mindset for this project and not necessarily directly tied to it. And I know this is actually, I can't remember if this exact figure, but this has come up in previous VLMMLR talks from other speakers. I think, Sergey, you shared this where when you actually want to design a sequence for a conformation, you have to consider all possible states to make sure that you don't actually design a competing state. So this is all background. If there are questions, I'm happy to address them, but this is, I consider this just motivation for the project. P near is the numeric calculation for, I just went back with the slides. So this is the folding funnel that we just looked at. Very good folding funnels have high P nears, and poor folding funnels have low P nears. So this ranges from 0 to 1, where 1 is the best. When we design cyclic peptides at Menton, the P near calculations are, to the first approximation, all of our computational costs. It's so much more expensive to compute these funnels than it is to actually design our cyclic peptides. On top of that, it's actually very challenging to predict the outcome of a P near calculation ahead of time. Just to give a brief background, so to calculate these funnels, we generate 15,000 random conformations of the given sequence and relax and score all of them, which really adds up to a lot of compute. And it's not always, you can do simple things like prefer having prolines and other entropy restricting amino acids, but on top of the low hanging fruit, it's very challenging to tell if a conformation will have a high P near or not. So just from a development and business perspective, like we just save a lot of money if we could prune out some of these structures. If we could just predict if a P near calculation is worth running. We don't necessarily have to predict if it's good or bad, but if it's at least maybe good, that's good enough for us. Part of this is because the vast majority of P near calculations return something on the right more than the one on the left. So if we could just remove a lot of the false negatives, that is worthwhile. So we set out to learn as much about this as we could. We generated a bunch of data to perform machine learning. So we randomly generated 10,000 cyclic peptide sequences between 6 and 10 loop lengths between 6 and 10. So the largest cyclic peptide was 10 residue, smallest was 6. We had 50 different amino acid types, although this is largely double counting because there's L and D. So it's roughly 25 amino acid types with both chiralities. For those who aren't familiar with as non-canonical as much, so proline, of course, really favors P near by its rigidity. It implicitly disfavors alternate conformations. Hydroxyproline is just like proline, but more soluble. I forget what AIB stands for, but it's basically it's glycine, but if you take both of the hydrogens on the C alpha and replace them with methyls. So it's also if you overlap L and D, alanine on top of each other. So it's a very restricted amino acid from a backbone entropy perspective. So all three of these are considered backbone entropy restrictors. So we generated a bunch of sequences, but favored these to just biased towards the more fruitful area of sequence space. We measured p-near for all of these. So for a given sequence, we'll actually end up with 10 to 15,000 samples. When you compute a folding funnel, you can actually, like it's by one, get 15,000 free, because you can then compute p-near for all of the other points in the funnel, just by recalculating RMSD to put them at 0. So we ended up with 16 million samples. The sample is a cyclic peptide conformation with sequence conformation to use as training. I guess sometimes I lose the forest through the trees. So I should say the goal of this project is we input this on the left, right? It's a cyclic peptide conformation. And we're training, which we'll talk about in the next slide, a classifier just to predict, is this a high fold propensity cyclic peptide or a low fold propensity cyclic peptide? The architecture and stuff we'll get to soon. I just wanted to quickly, I don't want to take it for granted, even though a lot of us might have seen this before, but the graph neural network of a protein, you can do atomic level graph neural network. So each atom is a node, and then atoms that are bound are edges. I've only ever done residue level. So for example, here we have a cyclic 8-mer. Each residue is a different color that corresponds with the nodes. So one node is a residue, and then we connect edges between residues that are close in space. For this project, since we're considering the current conformation, but all possible conformations, we're actually doing a fully connected graph. But the conventional method is residues that are close in space get edges. So just as a bit of background. So all-to-all graph in this case. Again, we can gloss over this if you're not as interested, but I like to share this, especially for, I don't know, I always like to see this when people present, but our model had, so this is entirely just a graph neural network. Graphs have X tensors, which show the node features. Circles are data, and then squares are layers. So the input data, there's an X tensor for node features, an adjacency matrix, and then edge features. Then the output data is the prediction. I do something weird, and I hope people can call me out if this is a terrible idea. I don't normalize my data at the NumPy level. I insert it into the network unnormalized. And then the first thing in the network is a batch normalization layer, which gets frozen after the first epoch. To me, this is quality of life with no real loss. But I don't know. My benchmarks show it's scientifically the same as normalizing ahead of time, but I'm open to being corrected on that. Nodes, edges, pre-process with a single dense layer, go through 3 graph convolutional operations. XCNet is a convolutional layer that we put together at Menton a year ago. Oops, I'm sorry. I slammed on the desk so hard, my other hand clicked the mouse. The output of, so XCNet is relatively, one thing about it, it updates edges and nodes, where most graph convolutional networks only update nodes, which is why we like it. However, after the final one, only the nodes survive. So we concatenate the input nodes with the process nodes dense layer. This is a circle. So this just means that we named the data at this point, saying that it's just the final node representation. Because then we undergo a global attention pool. So essentially every node in the graph gets summed together with an attention operation. So it condenses down into just one tensor, one big node. That goes through a few dense layers, out comes your prediction. So this is maybe in the weeds for some people, but if you're new to GNNs, I always like talking about this kind of stuff. Okay, so that's all the setup. rest of the talk, I believe, is either data or forward-facing, like Rosetta things. Okay, so
Jack,
hyperparameter.
could you, I think I might have missed it, but what exactly are you using for the loss in this case? So you have a prediction, and what loss function are you using for your
Yes,
optimizing?
this is just binary cross entropy. So the output is a 1 if the input graph represents a high p-near cyclic peptide, and a 0 if it's a low p-near cyclic peptide.
Got it. So you're not like trying to output the actual p-near value or anything like that, or?
Right, this is just classification.
Okay, got it. Thanks.
So this to me is the fun data, and I hope it's not jumping the gun to just go straight into the feature thing, but you know, we trained the model many times with many different, I guess, completeness of the graph representation. So for this first box and whisker, these are 10 different models. These are the binary cross entropy losses. However, for these models, the graph that is input into the model only represents the cyclic peptide backbone. So phi and psi angles, you know, distances between backbone atoms, but no information regarding the amino acid identity or the sidechain conformation, or even sidechain interactions in any way, or sidechain backbone interactions. This is somewhat a control. We didn't really think this would be the best, but it sets the stage. The next box and whisker, the red one, is a superset of the previous one. The only addition is that it labels nodes if they're L or D amino acids, or if they're that one special achiral AIB amino acid. So now we're starting to cheat, because for one amino acid, it is explicitly labeled, but every other amino acid is unlabeled. So a little bit of hand waviness, but for the most part, this is only considering the backbone conformation and chirality. And just by saying L and D, you see a noticeable increase in trainability. I think this is largely just from a backbone entropy perspective. Like, if you can, once you know your Ramachandran plot by knowing your chirality, then you can further assess your backbone conformation and know if it's stable. Green is another, so this is, again, building off the previous one. So a superset of everything shown up to this point, but now every edge and every node has the respective Ref 2015 term. So I forget the exact number, but there are a little over a dozen terms in Ref 2015, and each one gets represented directly on every node, and so that one body energy is on nodes and two body energies on edges. And it's as presented in Rosetta. So hydrogen bond, backbone, long range, backbone, short range, backbone, sidechains, you know, all of the individual terms in the weights file of Ref 2015 gets its own feature. And that performs the best. The final one is, so this is where we break the superset pattern, because instead of adding Ref 2015 energies, we build off of red, but we add the amino acid identity and the chirality, or sorry, the chi angle of the sidechains. So in theory, all of the information required to rebuild every atom of the entire cyclic peptide. So what's interesting, so not only is this a superset of red, but it is, you know, in theory, green is a function of blue. So whatever you can learn from green, you can learn from blue. I believe that this performed poorly due to overtraining. I'm not convinced that this could not have done better. Maybe a more skilled, you know, or just luckier person might have gotten a better score with blue here. But at least out of the box, these are the comparisons. So again, asterisk by all of this, but I just from a skill perspective, but at the level that I can do this, Ref 2015 outperformed the other representations. I'm also showing the ROCs on the right. and I'm showing the best one for each model, and I'm going to zoom in. So sorry, let me do that slower. So we have 4 lines. So the best line from each class is shown. And I'm just going to show green and blue, and I'm going to zoom in on the top left. So we go from 0.5 to 1 and then 0 to 0.5. I also, as a reminder to myself, so false positive rates determine runtime. So from a practical perspective for us, we're very interested in decreasing false positive rates. And so this point for my coworkers and I really stood out because to the first approximation, 0.2. So it speeds up our entire pipeline by 5x, but we get to keep 95% of our true positive hits. So I have definitely been caught in the past of comparing features between models that is comparing bad against worse. However, I believe that these, at the very least, it's good against good. I mean, from our perspective, this is a successful training run. So all of this is just to say that I don't believe this is bad against worse. This is actually comparing good models against each other.
Jack, can I ask a simple question? I'm sort of new to the deep learning side. Why wouldn't you train the backbone chirality AA chi with the input from the backbone chirality, like start with that network, or is that a possibility? So it would only get better from that point, or is that?
Yeah, that's really interesting. So, so if I understand, correct me. So you would start with one of these red models, and then re-architecture it to include these features.
Yeah.
You train. I have no reason not to do that. It actually would be a great idea. I don't know how.
Because I'm assuming you grew the network when you added these additional features. Like, is it easy to snap that portion network and add more layers, I'm guessing?
So all of the networks shown here were grown from scratch, like they were trained. So like the green models.
I'm just trying to understand how difficult it is, rather than training them from scratch, to train them with models that you had trained in a previous step.
That's one of those things that's doable. I haven't ever. I'm sure that you could find some fruit there. I just.
I guess if you could technically zero out some features and train the model and then sort of enable them again. So that way you don't really have to change anything. Like in this case, the only thing that's changing is what you're inputting to the model. So initially you could zero out the chirality and so on.
Yeah.
Exactly. I think sort of related to this question, Jack, you mentioned that maybe what's happening here is that you're maybe overfitting. That's why maybe the blue one's doing worse. Does the actual the training loss, does that get lower with the blue? Is it?
That's a great question. I should absolutely look at that.
Because if that one drops significantly, then that might actually indicate then that's maybe overfitting. Actually, then that might explain.
I believe that so it's been a little while. My memory is that all of these overtrain almost immediately. In fact, I have to break epochs into increments of 50 so that I could stop early if I have to. My memory is that the training loss for all of these was pretty similar. And the training loss didn't even begin to flatline by the time we killed it. So my memory indicated that this was an overtraining case, but it's rusty enough that I can't answer your question with much confidence. But that's a great point. I should look into that.
Mm-hmm. Yeah, that is interesting. Okay, thanks.
It is tricky too, because we have millions of different confirmations, so we thought that we'd be safe, but they're all derived from only 10,000 different sequences. So I think that we're kind of using the number that makes us more comfortable instead of the smaller 10,000 number, which we could overtrain on. So, because we have many different confirmations from the same sequence. But that is, it's a great thing that I should look into. This actually just came out very recently, but we wanted to see what the model looked at. And I'm very open to folks telling me that I have the wrong, I could like do this better. However, in the meantime, we're looking at the Rosetta terms, and we're taking the partial derivative of the prediction with respect to the input feature for each individual input feature, and then adjusting it, like rescaling it to accommodate the standard deviation of that input feature so that the scales are comparable. So essentially we're looking at what is the magnitude of the gradient for this feature with respect to the scale that we expect this feature to change from sample to sample. Most people don't have to clarify that because they normalize their data, but I'm slowly learning I should use best practices instead of my quirky way of doing it inside the model. However, mathematically it's the same. So all that's to say, what we're plotting here is that essentially we're plotting the gradient of the output with respect to the input. You take the absolute value and you mean over that. So hopefully conveying the impact of each feature. Some of these are 0 because they don't actually change from sample to sample. FALEC doesn't have a meaningful, like, it's always 0 for a one body energy. So that's always 0 here. This essentially, these are just a bunch of different biases or bias nodes in the training context. And in theory, we could have removed them altogether. I kind of, from a purity perspective, I just added everything to every node and every edge because I didn't really know if there were edge cases in Rosetta. But so worst case scenario, it's just useless and a waste of space. But so that's all to say, that's why there are zeros. And there are more edges in a graph than nodes, which is why I believe the edge features are somewhat smaller. So this is a per node scale and a per edge scale. Nodes really like looking at the phi angle of the backbone. PAAPP is the probability of an amino acid at a position given its phi and psi value. This is a Ref 2015 term. Then this one here is, this labels positions that are proline, hydroxyproline, or the D-proline, dihydroxyproline. So it's not purely sequence agnostic. We only really make explicit special cases for the backbone entropy positions. On edges, they really like looking at FA attract, FA elect. Leonard-Jones attractive potential and the Coulomb potential. Also the salvation term and FA rep, the other Leonard-Jones side is up there. For the most part, everything is relatively flat. Same with the nodes, the smallest ones. So like YHH planarity measures that, like in a tyrosine, the hydrogen at the tip of the tyrosine is in plane with the ring of the tyrosine. And since most of these are Rosetta generated, like this is almost always 0 or close to ideal. And then the smallest one over here is the short range backbone hydrogen bond term. I think that's small just because it's redundant with something that we explicitly labeled. So backbone H bonds here, which is funny because we explicitly labeled it because it's very important. So the smallest one is actually important, it's just elsewhere. So this we just thought was interesting to look at what the model is looking at, and so cycle size actually didn't play that large of a role directly, even though we thought that'd be a big deal. Like 6 mers is much easier to have a high p-near than a floppy 10 mer.
Yeah, why would you want cycle size in there? Because I mean, that's not some, because you don't want to just know that if it's a 6, it's more likely to fold or less likely to fold, right?
We wanted to give all the information we could. In theory, the graph could figure out its own cycle size. So we didn't really think that it was cheating. It was just sort of saving the graph some time.
But a model that just tells you that it's better to design smaller proteins with more prolines in it is not really
Yeah.
that informative for what you want, I assume.
Well, because again, what we're looking at here isn't really the end goal. This is really just an insight into the reverse operation. So what we want is something that can just take a cyclic peptide and by any means necessary classify it as high or low pner. And if it has to just count prolines and that's good enough, then that's fine by us. You bring up a good point though, like this maybe didn't require a neural network to learn that prolines help. So again, so this slide is really more insight than end goal. So this is sort of part 2 of 2 in terms of analysis. This Rep 2015 representation is pretty special in the sense that it doesn't explicitly label, like it doesn't one-hot encode amino acid type. In theory, we could pass in a cyclic peptide that has a new amino acid type and we would not have to retrain the model. Even though that technically works, we want to test to see if that practically scientifically works. And so this next test is the generalization test. Can this model still work if applied to peptides containing amino acids absent from the training set? Just to explain how we manage the data here. So I'm getting a little bit of microphone feedback from somebody. I don't hear it anymore though, so thank you. So when we originally trained our models, we had, and forgive the overly simplified, like this rectangle is all of our data, and then we split it into training and validation. We then wanted to test, we're using DAB as so this is a non-canonical amino acid that we want to test for this. We split all of our data into 2 sets, or further into from 2 to 4 sets. So if DAB is absent from the cyclic peptide sequence, it's over here. If all the cyclic peptides that contain DAB is over here. We then train new models on the new training set where DAB is absent, the new validation set where DAB is absent, and then compared the performance of this new model against the original model on this corner, where DAB is present. DAB itself is very similar to lysine. We sometimes just call it stubby lysine because it's just shorter. It's this middle one. So DAP is a very short lysine. DAB is, there's, I believe, 2 triangles. Orn, 3 triangles, lysine, 4 triangles. Sort of a Pokemon style relationship here. And the model trained without DAB is shown in yellow. The original model that was trained with DAB is in orange. All of, and then we measured the ROC exclusively on data containing DAB. Exclusively on data containing DAB. So the fact that these 2, and we're plotting the area under the curve. So the fact that these 2 boxing whisker plots are roughly the same distribution shows that there's actually no loss, to the extent that these are the same distribution, there's no loss in predictive quality. We can successfully operate on new amino acids using this Ref 2015 feature set for our graphs. The ROC curves roughly overlap with one another. Again, we're showing, just like in the larger plot before, we're only plotting the ROC for the highest AUC, so like the very top model of each. So perhaps the average one is worse, but at least at its best, they can compete with one another. And this was a huge win. We wanted to repeat this. So the big old letters on the left are the amino acid being held out. So for example, here, yellow, trained in complete absence of glutamine. Orange, again, is just the model from before. And we're plotting the AUC on the data exclusively containing glutamine. And that's actually, so that's why the orange spots are different. So these orange 10 are the same 10 neural networks as these 10, but the y-axis is different because the data is, this is the glutamine data, this is the histidine data. And so we see consistency, same predictive ability, at least by eye. You could say this one's maybe a little bit higher, but I'm nervous about that because, I mean, I don't want to be overly generous because then I have to be less generous here when we're a little bit lower. But overall, sorry, so the previous one was polars. Here we have some hydrophobics. You can see the data is a little bit more sparse, but the pattern still holds. And the conclusion really is that we don't have to retrain our model when we introduce a new amino acid chemistry to our design architecture in Menton, which is great. It means that we don't, it's very expensive to collect data on the new chemistry, and you have to retrain the model. And the fact that we just don't always have to retrain, I believe this is a huge win, both for us in the small sense, but also it shows what Ref 2015 is powerful of as a neural network feature set. So this is really my last slide on the actual science of this. I do have one more slide just talking about how Rosetta could change code-wise. I'm also double dipping because I'm advertising a preprint that's hopefully coming out soon while also using the authors of that preprint as an acknowledgement slide. And the title is a conclusion. So our conclusion was that Rosetta energies as neural network inputs do allow for sequence generalization, at least in this classification project. And so Shannon in the Myler lab helped out with this during her Menton internship. And then Pat, Stephen, Hans, and of course Vikram, who are all coworkers at Menton. Although, and then Vikram has other hats, have all helped with this and come to this conclusion. We are hoping to get this out the door by the end of the week. So hopefully this will be readable soon. And I'm also very open to if you don't think the data really sells the narrative that that I'm pushing, because if I can't convince you when I talk about it, then I'm not going to convince anybody when I write about it. So yeah, and with that, I'll take any questions or thanks for listening, and I'll just mumble until I see a hand or.
Jack, I got a question.
Sure.
If you applied this to your input data set, your 10,000 designs, what do you see what features do you see enrichment in the output models? That would pass?
If I applied this to the data set.
I'm looking at what you found as the input. I would guess that you would find small proteins with proline rich and hydrophobics in the core are things that are more likely to pass the p-near. And so I'm wondering if you do see that, because you can now apply this, I guess, to your training or your test data set of 10,000 proteins and see enrichment for certain features.
Exactly. Yeah, and it is sort of funny because it's all this stuff that you'd expect. We like seeing small cycles. We like seeing proline AIB. We like seeing backbone hydrogen bonds. You could probably argue that this could be accomplished with some sort of random forest that just, takes a very small number of features and, just takes what we already know to expect.
Well, I think this type of work is always the most interesting when you find things that you aren't going to expect in it. So if you ran your data set through this algorithm and then said, okay, these are the things that we would expect to pass, but didn't pass. What are the small selection that did pass but has unexpected features, like doesn't have any prolines in it? What are the ones that are passing that don't have quite as many hydrophobics? What are other things you can tease out from this that are the models picking out that are really good, but you wouldn't have expected them to be good?
Great question. We mostly did that in the other order, or in the other direction, because we're a small new company. We only really have enough data to make anecdotal. We learn by anecdotes, and so we picked up some like perceptions that didn't hold true. So before this experiment, we thought that if you only had 2 prolines, you'd want to have them on opposite ends of the cycle. And from this experiment, we really found that that doesn't matter. 2 prolines can be anywhere on the cycle relative to each other. And from a data perspective, we don't see any change in signal. The same is true for, like we looked at different chiralities or chiral patterns, and we didn't really see any, we had the hypothesis that maybe there's a magic sequence of chiral, like LLDDL or something like that, and none of that rose above the signal, or none of that signal rose above the noise. And so really what we really just disqualified personal hypotheses like that, then found new hypotheses.
I bet there's a new hypothesis in there somewhere though.
There's gotta be.
It's good stuff. You'll probably like 6 months from now, when you run this on another 10,000 designs, been like, oh, look at that statistic.
Exactly. Yeah, I do hope that we find new things out of this.
I have one other unrelated question. I've heard of p-near before. Is there a script for that that's been published somewhere? Or is it in Rosetta somewhere?
It is.
Is it part of Chris Norn's paper? Or the Smith paper?
Well, it's definitely in the Smith paper. I believe it traces back to Vikram. I don't know exactly which paper. Let's see. It's buried in protocols. So I believe it's protocols, cyclic peptide predict, no, SIMPSIC.
It's from Patrick Conway from a long time ago.
Yeah, is it
To
a
measure-
script?
It's to measure how funnily a funnel plot is. And Vikram's thing, I think, trained on p-near to be able to assess how p-near-ishness without having to do all the forward folding.
Right. That's so, but the p-near script that you pass in a funnel is in Rosetta. It's in protocol somewhere.
Yeah, I forget how it's abbreviated. It's something like simple stick pep predict.
Okay.
It's an application in protocols. Sorry, it's also an application in applications. It's yeah. If you grep those words, it'll pop up.
Okay.
I can send it to you after too. I'll post it in Bloomler.
Nope. Somebody posted it. Oh no, that's an article. Never mind.